Hi, we will continue to next one. Okay, so the development of zygote. Okay, so previously we have seen the menstrual cycle. Okay, menstrual cycle. So that means after the cycle, so we will focus on ovulation. So if ovulation or after ovulation there is fertilization. Okay, there is a fertilization. That means we will have a zygote. A zygote is formed after fertilization. Okay, so that means we will start the development of zygote from fertilization. After fertilization we will see how the zygote is formed, okay, or where. Okay, then after the zygote form, okay, we will see the next steps, okay, where the zygote will, the parts of the fertilized zygote, okay, some of the part will develop into the placenta, and we will see what is placenta, that you have know. Okay, you know placenta, and then you know the function, but do you know how it's formed? Okay, so this is the part where we will see, we will we will see what is placenta and what are the content inside. Okay, and then we will see, is it similar to what you have understand before or not. Okay, then number 4 part or the objective, okay, we will see the importance, okay, of the circulatory system. Okay, to know that, okay, to know that the blood, okay, in the baby, the fetal means the baby, yeah, okay, and the mother, okay, they are separated. Okay, they are not mixed. Okay, there are no mixing of blood between the fetal and the mother. Okay, so this is why we need to have that. Okay, later we will see. And then also the last part is to see what or how the identical twins and the fractional twins is formed, okay, or is produced. Okay, how to get. Okay, then Siamese is another one. Okay, Siamese we will use the word conjoin. Okay, conjoin, but your book use this one also. For temporarily we will use this. Okay, but it's better to use the word conjoined thing. Okay, before we start, we see a video on the journey of the sperm. Okay, a sad story of it. Okay, how one sperm success, okay, to fertilize the ovum.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life. It is the story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, an opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells from the woman's immune system, mistaking the sperm for foreign invaders, destroy thousands more. Next, half the sperm head for the empty fallopian tube, while the other half swim toward the tube containing the unfertilized egg. Now, only a few thousand remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At long last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the corona radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
So you can see, not easy to fertilize an ovum. So the millions of sperm, has get sacrificed in order to fertilize one ovum. Okay, now let's start. So fertilization. So as we know, after menstrual cycle or the day after the day 2 weeks, about 11 days to be exact. The day 11 until the day 17. There's a period of time. Where the ovum will be released. The secondary oocyte is released from the ovary. When the follicle matures, secondary oocyte is being released, enter the fallopian tube. So when it move around in the fallopian tube, if fertilization happen, if the secondary oocyte met with sperm, that means it will continue the meiosis, until it has the matured nucleus to fuse with the sperm. So fertilization happen when the sperm fuse with the ovum. Later we will see a videos on how it been fertilized. So how it's being fertilized, how the sperm get to the ovum. And then we also need to know there are a few layers in the ovum. So this is secondary oocyte. The nucleus is here. So the minute the sperm you see the sperm bind to zona pellucida. This zona pellucida is the layer in the ovum. So the minute the sperm reach the ovum, the nucleus will complete the meiosis. And then the nucleus inside the sperm will fuse with the nucleus in the in the ovum. Okay, then it is fertilization. It is successfully fertilized. Okay, and then the minute it enter, okay, the other sperm, okay, you see the other sperm will be bounce away. Okay, so the bounce away, it will not go in anymore, and then you will see the same sperm here. Okay, it will only release the nucleus, okay, into the egg nucleus to join and become diploid. Okay, so haploid and then haploid become diploid. So when the sperm nucleus enter the egg cytoplasm, so number 4, you see, so only the nucleus enter the nucleus ovum. Okay, the nucleus of the ovum. Okay, so when they union, okay, they join together, so then we call them the zygote. Okay, so the zygote will begin with a single cell. Single cell means it's a diploid cell now because of the nucleus. See, the nucleus of the sperm plus with the nucleus of the ovum, and this is called one cell. Okay, the zygote is one diploid cell because now only he has the complete chromosome. Okay, before this is just half and half. Okay, from mother, half from the father. So after he forms zygote, it become complete. Okay, means it's one cell. Okay, with 46 chromosome, and then it will start to divide rapidly, the zygote. Okay, in the following fertilization. I mean, after the fertilization, and after a few weeks, okay, it become embryo. Okay, so from zygote it become embryo. Now let us see. So during ovulation, okay, it release the secondary oocyte. So after that, fertilization. So from fertilization, a few weeks, it will undergo mitosis. Okay, divide. So from one zygote, it become 2 cell. Okay, zygote is one cell. Remember. Then it divide into 2 cells. After that, of course, 4 cells. These are all mitosis. Okay, 2, 4, 8. Okay, there's no 6. There's no 3. Okay, this is mitosis. Okay, if you understand mitosis, you will know that every mother cell will produce 2. So here 4 cell, the 4 cell will become the mother cell and produce 2 each. So it will have 8. So 8 will produce. Okay, 8 every 2 will be 16. Okay, after that, and so on, 32 and so on. So it will be very, very fast until a stage where we call it morula. Morula is made of 16 cells. 16 cells, then after that start to implant. Okay, it will find, when it reach morula, it will start to find a place to implant. So let's say the morula never implant here, okay, at the uterus lining, then it implant in the middle here. I mean during the at the fallopian tube. I mean during it the movement. Okay, then we will call it, this is a pregnancy, ectopic. Okay, ectopic pregnancy. So it's not good. It shouldn't be in the fallopian tube. Okay, it should implant at the uterus. Then it can develop, okay, into a fetus. Okay, now from morula, it needs a few weeks. Okay, a few days. Okay, to become a blastocyst. Okay, about 100 cells lah. Okay, when it reach about 100 cells, okay, we call it blastocyst already. Okay, so the number of cells is still okay. Okay, you no need to memorize that. Okay, just to know that from morula, it will end up with blastocyte, and then this is the part which is very important. So after that, blastocyte, it must implant. You see, it must implant. So let us see. Okay, yeah. So there are 2 stage, 2 cell, then 4 cell, then 8 cells. After 8 cell, you have 16. Okay, 16 cells, 64 cell. This is also morula. Okay, after that, when it less than 100, we call it blastocyte. Okay, now this blastocyte or blastocyst, okay, inside, okay, outside is one layer, inside they have one mass. Okay, we call it the inner cell mass. Okay, this inner cell mass, okay, are the one that will develop into embryo. Okay, and then the outside one, okay, they will form the amnion. Okay, the amnion. Okay, then part of it, okay, part of the blastocyte, okay, the cell mass, it will become, part of it will become placenta, part of it will become the embryo. So you have to know lah. Okay, later we will see in detail. So after implant, okay, this one is to show implantation. Okay, after implant, okay, the blastocyte start to develop. Okay, develop means it undergo differentiation, okay, mitosis, division, to separate the layer of cell into a few layers with different functions. So you see, we'll see trophoblast here. Okay, trophoblast is the red one. Okay, blastocyte, the outside layer is called trophoblast. Then the blastocyte inside, inner mass. Okay, the trophoblast will become the membrane. Okay, the amnion. Okay, then the zygote will travel down the fallopian tube where it become morula. Once you reach the uterus, the morula become blastocyte. So this is explained. Okay, the blastocyte then buried into the uterine. Okay, the uterine wall or the endometrium, and then the process is called implantation. Okay, now we'll see short videos on how the development of the zygote. Okay, then only we continue with implantation.
This animation will show how your baby develops during pregnancy. Click the navigation arrows below the animation screen to play, pause, rewind or fast forward the animation. Pregnancy usually lasts about 9 months or 40 weeks and is divided into trimesters. The first trimester is from week 1 until week 12. The second trimester is between weeks 13 and 26, and the third trimester is from week 27 until birth. If you become pregnant, your womb doesn't shed its lining as it normally does at the end of the menstrual cycle, so your periods temporarily stop. This is because the egg, which has been released from one of the ovaries, has been fertilized by a sperm, and you are in the early weeks of pregnancy. Here we show the ovaries, the fallopian tubes, the womb and the vagina. The fertilized egg has already started to divide in the fallopian tube. It will continue to develop and grow in the womb and is called an embryo at this stage. Here we show the embryo in the womb. By week 9, the embryo has developed and is called a fetus. At this point, most of the organs, including the nervous system and heart, are forming. Here we show the fetus in the womb. By the time you are 12 weeks pregnant, your baby is approximately 6 centimeters or 2.5 inches long. At this stage, your baby's neck is uncurling and the limbs are complete. The eyelids are still fused and the ears are forming. Here we show the baby, the umbilical cord, the placenta and the amniotic fluid. The umbilical cord connects the baby to the placenta. This is how your baby gets nutrients and how waste, such as urine, is removed. In the second trimester, your baby's sex organs develop and other organs mature. The baby swallows amniotic fluid and passes it out through its digestive system. The kidneys start to work and pass small amounts of urine. Your baby can now hear and is covered in fine hair called lanugo. Here we show the baby in the second trimester. You may first feel the baby move in the second trimester of pregnancy. The movements become much more vigorous and obvious as the baby gets bigger and stronger. Braxton Hicks, or practice contractions, are painless tightenings of the womb that may start from around 20 weeks of pregnancy. Your baby's lungs mature throughout the third trimester. The baby makes breathing movements even though the lungs don't work properly until birth. The baby grows fine hair and fingernails. The eyes open and close, and teeth may start growing under the gums. Your baby's fat stores also increase in preparation for birth. Braxton Hicks contractions become stronger in the 3rd trimester. At the end of your pregnancy, your baby changes position so it is ready for birth. The baby's head may not engage, that is, move into the position in the pelvis for birth until labour. This is the end of the animation. Click on the animation screen to watch it again.
Okay, now continue implantation. Implant, okay, you see, we zoom here. After implant, okay, see, just now red, okay, we remain, the red is still trophoblast, okay, trophoblast. So now the inner cell mass, okay, what happened to the inner cell mass? You see, this is the endometrium uterus, the blue one, okay, it means this is the mother, okay, this part is the mother, this is the baby, okay. Now you see, this mass, this inner cell mass, okay, they will have 2 parts later, okay. The parts of a part of it, it will become the yes, here one, another diagram, so this is endometrium, so this is trophoblast, so this is the cavity, so we will see this. So this blastocyte, the outer and the inner, so you see the blastocyte, inner and then outer means nearer to endometrium. The outer one, it will form with part of the, it will join with part of the trophoblast, and we call it the trophoblast. Then the inner one will become embryo, okay. So the outer one, okay, the outer layer of the trophoblast will become this part, okay, the placenta, okay. It develops into chorion. The chorion will surround the embryo, and then it develops into placenta, okay. So the trophoblast, at end of the day, it will become placenta, and then we will see the placenta is actually, this look like this, okay. The whitish color one is the amnion sac, okay. The one with this one, okay, a lot of blood one, okay. Those are the tissue which is the placenta. You see here, it's actually made of blood and blood vessel and capillaries, okay. So those are placenta. So this is placenta. So placenta is just blood, okay. Yes, the placenta will consist of the fetal capillary and the mother arteries and vein. So you will see this is the umbilical cord, okay. Umbilical cord is this one, okay, the one that join to the placenta. From the baby join to the placenta, that is umbilical cord. So from the umbilical cord, you see this blue and the red nearer to the umbilical cord, that is the fetal capillaries, arteries and vein, and then it branch into smaller capillaries. Then you see this side, okay, this side is maternal. Maternal means the mother. So the mother blood vessel will also enter the placenta, okay. So inside the placenta, it will just it will be the pool of blood, okay. So you can see here the darker pink color, this side is contain a lot of mother blood, okay, the blood of the mother in the pool, okay. It's like swimming pool, okay. So then this swimming pool, it will diffuse whatever nutrient into the capillary of the baby, and the waste product from the baby will enter into the mother blood, okay. Okay, so when it is in this dark pink color, so the blood of the mother, okay, so they will have higher concentration of nutrients, so the nutrient will just diffuse into the baby capillaries or the fetal capillary, and then the baby will get the nutrient. As for the baby waste product, it will diffuse also from the capillary into the mother blood pool, and then carried by the vein, okay, the maternal vein, and then go back to the mother circulatory system. So you can see here, okay, there is no mixture, okay. The mother blood is only at this pink color region, okay. Then the baby blood is in the capillaries. So there is no mixture of blood, okay, between the mother and the baby. This is very important, because sometimes the baby doesn't have the same blood group with the mother, okay, because we will see in the next chapter 5, chapter 6, okay, when the mother doesn't when the blood is not the same group, okay, it shouldn't mix, okay. That's why when the develop of the placenta, it is developed in such, okay, the mother blood will never ever mix with the the baby blood. Okay, they will have diffusion. Okay, and secondly, they will avoid a lot of disease that being sent into the baby. Okay, of course there are certain chemicals or certain medicine that can diffuse into the blood. Okay, but since it's not mixed, okay, it reduce the chances, or it reduce the amount of harmful substances entering the baby. So this is a way of protection, okay, for the mother, and also a way of protection to the baby. So their blood doesn't mix. Okay, so very important. Okay, now you will see here again. Okay, I need you to very clear on this. Okay, because it's quite hard. Okay, it's quite new to you. So you see, this is the umbilical cord. So in the umbilical cord, you have the vein, and then the capillaries, arteries, so all branch in. You see, so the blue, the green and the red is the baby. Okay, then the blue, okay, here, see, this is the maternal. Okay, and then this red parts, okay, this side is actually, you see, okay, this is uterine muscle, this is lining of uterus, so the fetal chorion, so the chorion is this part. Okay, so it's made from the trophoblast, remember. Okay, so this part is where the blood of the fetal. Okay, this part is the mother. So you see the pool of maternal blood, you see this one, here, the one colored in red. Okay, these are the pool. This one, the darker pink color, just now we mentioned. Okay, those are the pool, that swimming pool. Okay, the mother blood will flow inside. See, it will just flow inside, diffuse in the nutrient, diffuse into the baby capillaries, and then waste product diffuse into the maternal vein. Okay, and then it will remove, okay, by the mother. Okay, so this part is the placenta. We zoom out. Okay, zoom in. Okay, so again I will have to emphasize here. So this is another what we call a diagram. Okay, I want you to be very clear on this. Okay, maternal artery and vein. You see, there is no mixture. Yeah, you see, he go into the pool. Okay, swimming pool. You see this one. Okay, you see the baby capillaries. Okay, so there is no mixture. Okay, they are in two different circulation, so they do not mix. Okay, they do not mix. They do not mix, so the blood doesn't mix. So this is the fetal portion of the placenta, this is the mother portion. They have overlapping of the placenta, okay, but they do not mix the bloods. Okay, the overlapping part is just for diffusion. Okay, so the oxygenated down here, okay, what are being carried in,
So
and
now we see how the placenta is formed before we go into the twins. Okay,
Development of the placenta begins during implantation. When the blastocyst adheres to the endometrial epithelium, trophoblast cells differentiate into an inner cytotrophoblast layer and an outer syncytiotrophoblast layer. As the cytotrophoblast proliferates, newly formed cells migrate into the syncytiotrophoblast and lose their cell membranes, forming a rapidly growing multinucleated mass. The cytotrophoblast secretes proteolytic enzymes, and the syncytiotrophoblast sends out finger-like projections, allowing the blastocyst to embed into the endometrium. Lacunae, or spaces, begin to form within the syncytiotrophoblast. As the syncytiotrophoblast erodes the endometrial blood vessels and glands, the lacunae become filled with maternal blood and glandular secretions. Isolated lacunae fuse to form a network through which maternal blood flows, thus establishing early utero-placental circulation. Toward the end of the second week, small projections of the cytotrophoblast begin to expand into the syncytiotrophoblast, forming primary chorionic villi. Early in the third week, extraembryonic mesoderm grows into these villi, forming a core of loose connective tissue, at which point the structures are known as secondary chorionic villi. By the end of the third week, embryonic blood vessels have begun to form in the extraembryonic mesoderm of the secondary chorionic villi, transforming them into tertiary chorionic villi. Cytotrophoblast cells from the tertiary villi grow toward the decidua basalis and spread across it to form a cytotrophoblastic shell. Villi that are connected to the decidua basalis through the cytotrophoblastic shell are called anchoring villi. Villi growing from the sides of the stem villi are called branch villi. These branch villi are surrounded by the intervillous space and will serve as the main sites of exchange between mother and fetus. By the 4th week, fetal blood flow is established. Poorly oxygenated blood is transported to the placenta from the fetus via paired umbilical arteries. These arteries send branches into the chorionic villi and divide into capillary networks at the terminal ends. Carbon dioxide and waste products are removed from the fetal circulation across the placental membrane to the maternal blood in the intervillous space. Oxygen and nutrients are transported across the placental membrane from the maternal blood to the fetal capillaries. Oxygenated fetal blood travels back through veins, converging to form a single umbilical vein that goes to the fetus. Blood returns to the maternal circulation through endometrial veins, forced out by the pressure of incoming arterial blood. The placenta nourishes the fetus throughout development and is delivered shortly after parturition.
Okay, now twins. So we will see twins, how the formation of twins. So something that some of you know, okay, some certain things lah. So what we will need to clarify here, okay, so how it is formed. So when one egg and one sperm fertilize, okay, so there is a chances. So after they become zygote, okay, after they become zygote, they will start to divide, right? So there is a chance they divide into 2 separate cells first, then only continue with the next mitosis. Okay, when they divide into 2 complete cells, okay, they still have the same placenta, because this placenta is made of the ovum, the cytoplasm, the wall, the membrane of the nucleus of the ovum. So it still have the placenta there. Okay, but they have 2 cells, so they will have 2 separate umbilical cord but 1 placenta. Okay, so then we call this identical twin. But sometimes, okay, the ovum release 2 ovum, and then the 2 ovum is fertilized by 2 sperm. Okay, that means remember we have fallopian tube ovary, so the woman have a pair of ovaries. So normally they will take turns. Okay, every 28 days, okay, one of the ovary will release a matured follicle. Okay, but there are certain time, maybe under some situation, under some influence, effect, if something happen to the environment, okay, or the body of the mother itself, okay, that 28 days, the ovulation, both of the ovary release the ovum, okay, and then when that happen, okay, both is being fertilized, then we will have 2 different zygote, okay, 2 different zygote form, and then we will have 2 separate placenta. Then this is the fraternal twins, okay, or non-identical twins. There are many names for it, okay. So in your textbook they use the word fraternal twins, okay. So when you have 2 sperm and 2 eggs fertilized. So for fraternal twins, okay, the non-identical, okay, it developed from 2 eggs and 2 sperm, right? So it may have the same gender, okay, or it will have different gender, okay, for fraternal twins. But if for identical twins, because it come from the same ovum and the same sperm, okay, so only 1 ovum, 1 sperm, so it will either have boy or it have girls, okay, it will have same sex, the twins will have same sex, it will not have different gender, okay. Now this is the formation. Again, we summarize. So you just see here 2 and then 2 different zygote for fraternal, 1 zygote, then 1 zygote divide into 2 embryo, 2 cell. Okay, so from the 2 cell you will have the twins. Okay, but this one is it completely separate. Okay, let's say the embryo doesn't completely separate. okay, you will have what we call the conjoined twins. Okay, so conjoined twin is, you see, you can join at any part. So this example that I take from online, yeah, is the name of Kendra and Melia. So they have joined at the leg. Okay, that means the twins have only two. Okay, that means they share parts of the leg, but this is very considered lucky because they don't join the very important organ, so they can be separated by operation. Okay, but let's say it's a fatal organ. Okay, that means the very important organ like the heart. Okay, where they cannot separate into two. Okay, they do not have two heart, they only have one heart, then they have to leave it forever. Okay, like join together. So when it's not completely separated, okay, the embryo is not completely separated, that is how we will get the conjoined twins, okay, of the Siamese twin. So we will end here.
Before the slide end, okay, we will have one more short videos on the conjoined twins. So I hope you can enjoy it. Okay, so it's about conjoined twin. Then
thank you for your concentration. Okay, I see you again. Okay, thank you. Bye.
Twin sisters Lori and Reba Chappelle live remarkably typical lives, considering they have an unusual bond. They are conjoined twins, and though they share a significant portion of their brains, a common blood supply, and identical DNA, they still maintain their unique personalities.
We're so different that the only thing we have in common is that we love each other. That's about it.
Long a source of interest and fascination, this rare biological phenomenon occurs in only about 1 in 100,000 live births. Of these, only 1 in 4 will survive their first day. There are many different kinds of conjoined twins, each presenting unique challenges to survival and mobility. Where they are connected determines which vital organs they may share, the key factor in whether they can be separated after birth.
When I found out that the girls were going to be born conjoined, it was really a scary time, and they just said that your daughters are conjoined. I asked them where, and they said in the chest area.
Michelle Roderick and her husband Jeff fear the worst. If their girls share a single heart, chances of survival are virtually nil.
This is the liver joining the two infants.
Fortunately, this is not the case. The girls are linked at their breastbones, livers, and diaphragms. They are delivered by C-section without major complications, but there are worrisome signs.
Shauna wasn't doing so good. She was slower to come around.
She was smaller and having trouble. Say Janelle in this case would get really mad. She'd start just really throwing a fit. And when she cried so hard, she would actually push the surface blood off of her body and into Shauna's body. And Shauna wasn't crying, and all of a sudden she just would turn beet red. And Janelle looked really scary white.
The parents and doctors believe that twins should be separated as soon as possible. The chance of both twins surviving, 60%.
The overall surgery was about 7.5 hours. Separating the liver, it took them about 3.5.
The operation is traumatic, but the twins survive.
Once they were separated, they actually just would throw a fit because, they were used to somebody right there in their face, each other.
For some twins, like Shauna and Janelle Roderick, separation and intense reconstructive surgery can lead to great success.
I don't think it's very apparent that they were conjoined from the outward appearance. There are a few scars that only are visible if you look for them.
Ew, get off of here! She's like pulling my arm out of the socket. If we're still stuck together, that would be kind of weird. Like, my life would be all like, ugh. You wouldn't be able to do a lot of things. So happy that we're separated. I am, at least. You can do whatever you want to do, and you don't have to be stuck too hard.
When the risks involved with surgery are too high, however, twins must adapt. Such was the case with the original so-called Siamese twins, Chang and Eng Bunker, who moved to the U.S. from Siam in the 19th century. Attached at the abdomen by a 6-inch band of flesh, they stayed conjoined for their long, remarkably independent lives. Chang had a drinking problem. Eng did not. Eng fathered 11 children, while Chang fathered 10. Because of their shared brain tissue, separation was not a possibility for Laurie and Reba Chappelle either, and they too have adapted.
When I go on a date, she is non-existent. She sits there, she either reads or plays a game or does something. She just totally zones me out. I don't think about being a conjoined twin every day. It doesn't enter my mind unless something or somebody brings it up.
I am just an average person living on Earth, trying to do the best I can with what I have.
Robbers getting wrong.